we'll have any questions all related to during the presentation.
So thank you, Peter. And thank you everybody who's here and listening in. So we've done the system check. So as I mentioned, I'm going to be talking about RCrane. It's a Coot plugin for building RNA models into electron density. Before doing the demo, I just wanted to do a couple of slides showing you how RCrane works to give people an idea of what's going on behind the scenes with the plugin. Hopefully people use it and figure out what it's doing. So I'm sure most of you listening know RNA crystallography tends to be done at really low resolutions. There's a lot of existing RNA crystal structures. You see a lot of them are done in sort of the 2.5 to 3.5 angstrom range. We're lucky looking at maps that look like these. These are actually both model phase, so this is better than you're ever really going to get from an experimental phase map. And you can see from even these model phase maps, the only things you can really make out all that well in these maps are the phosphate locations and base. The phosphates are nice and electron dense. They show up very well. The bases are large and rigid and planar, and they're the things sticking out of the density for the backbone. So phosphates and bases isn't too difficult, but getting precise positions for really anything else is incredibly difficult on time consuming. And usually not terribly accurate. You know, there's placing some of the sugar atoms, you can see sort of generally where the sugar goes, but trying to place the sugar itself precisely is almost impossible when you're just staring at the density. Model building historically hasn't had very many good tools for model building or really good about doing it. A lot of it started out of you sort of stare at the density that's not all that specific, and you try to cram nucleotides, or you try to cram in stretches of nucleotides with less work, as I mentioned before, very time-consuming and very inaccurate. And what I've tried to do is add an extra step into the model building process, where you'd have a human being go from the density map, and with the help of the plugin, do this basic low-level trace of the backbone. So tracing just phosphates and bases. So as I said before, these are the things that are able to make out with an electron density. And from this trace, the plugin can calculate out very accurately and very quickly coordinate all of the backbone atoms. That this works is there's three different means of analyzing RNA structure. One is the backbone conformers, and one of those is the pseudotorsions, and the base phosphate perpendicular distance. And so I wanted to give sort of a brief introduction of what these three things are, since they're very key to the way that the plugin works. So the backbone conformers, you can think of these as the RNA equivalent of a protein side chain rotamer library. Here, instead of working with the side chains, you're looking at the backbone. So the consensus conformer library enumerates about 50 different preferred configurations for the RNA backbone, showing 6 of them here. They're given 2-character names. Typically, they're in a number and a letter. So 1A, for example, is the helical conformer. So that means standard A-form RNA helix looks like this, and it can be called the 1A conformer. There's other things you can see 4N and A. There's a couple things that I wanted to point out about these backbone conformers. The fact that they use the suite division of the backbone. So typically, you think of the RNA backbone, you think of nucleotides. The conformers use suite, so it's going sugar to sugar with a phosphate in the middle. Before people typically think about nucleotides, where you're looking at phosphate to phosphate with a sugar in the middle. So you can think of suites as being sort of half offset from a nucleotide. So one suite contains the first half of one nucleotide with the second half of the previous nucleotide. The other thing about the backbone conformers is that they're defined using approximate torsion values. They're not exact, so that means that if you had some structure that looked almost exactly like this, but there was one atom, you know, 1/100th of an angstrom out of the way, that still counts as the 4N conformer. It's the same way that the protein chain rotamer libraries work. They're all approximate values. So for one suite, you get values for delta, epsilon, zeta, alpha, beta, gamma, and the next delta torsion. And you'll see that there's 2 delta torsions per suite. The main important thing of that is that 2 adjacent suites overlap in their central in their central sugar. So that means one suite. This sugar is also contained in the next suite. So the next suite starts here and contains this nucleotide with the first half of the next nucleotide. One of the things of analyzing the RNA backbone that this plugin uses is the RNA pseudotorsions. So these are things that we've been using in the PIA lab for probably almost 15 years now. Behind the pseudotorsions is you simplify RNA backbone down. So I'm simplifying it down by looking at just phosphate and C1' atoms. In these atoms, you can define virtual bonds. I'm showing them here in red. I get two pseudotorsions, eta and F. And two pseudotortions. You can same things with them that you can do with, say, a protein Ramachandran plot. You now have two numbers that represent each nucleotide. So you can plot them in two dimensions, and you can look at this plot, and it can tell you things about your structure. In the past, we've used this a lot to analyze existing crystal structures. Here, obviously, I'm using it to build new crystal structures, but just give you some idea of where this means of analysis came from. I mentioned the base phosphate perpendicular distance. Basically a way of determining sugar pucker. So I mentioned before, if you're looking at density, typical RNA density, trying to pick sugar pucker is really, really difficult and many times just absolutely impossible, especially when you're working with experimentally phased data. So in the map, you can see that the sugar should go somewhere in here, but trying to figure out exactly which atom is out of plane is impossible. So phosphate perpendicular distance, you can figure out sugar pucker through this very simple metric. Your glycosidic bond, you extend it infinitely in either direction. That's what this black line is for, just a line drawn through the glycosidic bond. And then you measure distance from this black line to the 3' phosphate. So what you're interested in is the distance of this yellow dotted line. And if you take a data set of existing crystal structures, you can see that the data divides up very, very nicely. And this base phosphate perpendicular distance, or p-perp for short, anytime this p-perp distance is longer than about 2.9 angstroms, you know you're working with a C3' endonucleotide. And it's shorter than about 2.9 angstroms, you know you're working with the C2' endonucleotide. I should note that this technique is the same thing that's used in mole probity. So if you feed an RNA structure into mole probity, and it tells you have 11 sugar puckers that are questionable, it's using this same metric. And I can't credit for this. This was developed in the Richardson lab, and as you can see from the plot, works very, very nicely. So if we go to the slide that I showed before, this figure here looks pretty much like the pseudotorsions that I was just talking about. This figure here looks pretty much like the RNA backbone conformers. So the main thing with RCrane is it helps you place phosphates and bases, and then use pseudotorsion information, the p-perp distance information to predict and build the appropriate conformer. So the way that this prediction works is RCrane for every single suite. So let's say we're looking at this suite, it'll give you a list of guesses, which says the most likely conformer is 1A, and the second most likely conformer is 1C, and the third most likely conformer is 4A. So that gives me a really easy way to try to figure out, okay, how accurate are these predictions? I can set up solve crystal structures where I know what the conformer should be, feed it into RCrane, have it make guesses about the conformers using just phosphates and bases, and say how accurate was the first guess right, how accurate was the second guess right, how many times was the third guess right. You see that the first guess is almost always right, about 80 to 85% of the time. Usually when the first prediction isn't correct, it's the second or the third guess. You usually don't have to go any lower than that. Very quick proof of concept to show you that this tech at least can work. I built a couple of high-resolution crystal structures. So the green is the previously published coordinates. The ball and stick is what RCrane was able to rebuild using just phosphates and bases, so that the ball and stick hasn't been minimized against electron density. And you can see that this prediction methodology works very, very well. Got with this in the previous slide is I'm using published coordinates. So just to make sure that these published coordinates are going to be less accurate than you'd expect when you're first building into experimental density. So just to show that accuracy isn't going to affect the predictions, I read all the predictions after randomizing the coordinates to sort of simulate error that you could expect from experimental phased density. And you can see you really lose much accuracy from base angle and base movement changes. So basically rotating the base within the plane or moving it around. As long as you're within about 2 angstroms, maybe 1.5 to 2 angstroms, you haven't lost much accuracy. With phosphate coordinates, that does need to be a little bit more accurate, but as I mentioned before, phosphates are very electron dense, so trying to get them within an angstrom or an angstrom and a half of their correct location isn't a horribly daunting task. I show now just to give some sense of scale for this plot. 2 nucleotides in an A-form helix are about 2.7 angstroms apart. So if you're working over in this area of the plot where you have 3 angstroms of error, the nucleotides, that's where the next nucleotide should be, and this really isn't an atomically interpretable map. I just wanted to go into the demo and show how RCrane works, and you guys have some idea around and what it's doing. So this is 3.1 angstrom experimentally phased RNA density. We're here, the experimental phasing actually worked very, very well. But as you can see, especially used to looking at protein maps, this is just incredibly low resolution. To give you an idea of what this is supposed to be, here's a structure that was eventually built into this map. But obviously, you're starting out the building process, you're just going to be looking at this. So crane to trace density, trace the structure. The first thing I do is try to find something close to a phosphate, someplace where I want to start the trace. I've decided, okay, this looks like a phosphate, and then this would be the backbone. So here's another phosphate and more backbone. This would be, so I want to trace a nucleotide that goes from this phosphate, this base, up to the phosphate. Again, first I'd move my screen center. That's this little pink dot that you can see. Someplace to where I want to start the phosphate. I go up to the RCrane menu in Coot. I want to do a new trace. We'll look at phosphate coordinates, predict all the potential phosphate coordinates in the map, and then just try to select the one that's closest to where the screen center was. So it's important to get the little pink dot near where you want to start the trace. You can see right now RCrane is suggesting a phosphate coordinate right in the middle here. That sounds good to me, so I'm going to click accept. Now RCrane's going to try to trace the next nucleotide. You can see it's done things a little bit backwards from what I was aiming for. For the first nucleotide, it doesn't have any directionality information to go on, so it doesn't really know I want to be tracing up as opposed to tracing down. So it's traced something in the wrong direction what I want it to go, but I can correct that. I go over to the RCrane window and say next phosphate. So that's going to cause RCrane to try to give you a second guess for the next nucleotide. You can see going in the right direction, but it's swapped the base and phosphate positions. So I'm going to click next phosphate again. Okay, so now it's traced what we wanted to trace in. You see it's placed the phosphate, it's placed the base. That looks good to me. So I'm going to click accept nucleotide. Now I'll try to build the next nucleotide, and now it has some direction information to go on and be building in this general direction. So you place the next nucleotide right about where I was talking about before. So we're going this way with the chain. So that's me. I can click accept that nucleotide. I'm going to try to build in the next one. You've sort of gone around a corner here, but RCrane has managed to build the next base in correctly. So I could click accept nucleotide, but just to show you some other features that are part of RCrane, if I know the sequence that I'm building in, so if I know the portion of the molecule, I know I'm building in a GNRA tetraloop or some specific type of sequence, I can pick appropriate base type from here, change the base as I'm tracing. There's structure in the area. If you have some reason to believe that this base is syn instead of anti, you can click this and it'll flip the base. You'll notice not just rotating chi 180 degrees, it tries to keep the base roughly in the same position of space. The idea being if the nucleotide is already in the density, you don't want to move it out. So this flip button will adjust in. We're trying to keep it within the density. If for some reason you want to tweak the base position, you can click on manually adjust button. It'll open another window. You can start rotating things a little bit. This is real if you're tracing or if you know base, if you know via base stacking that you have some more information about the base plane. Sometimes you can tweak the base and get things a little more accurate. Again, that looks pretty good, so I'm going to click accept. This is basically the main of the tracing section is just RCrane will attempt to build a base, and you tell it yes or no. That's pretty good to me. I know since this molecule, this is a density for a base that's stacked. So this is what I want to have built. So again, accept nucleotide, trace more nucleotides. That looks good. Click accept. Got about nucleotides traced. And I keep going. You can see RCrane's managed to trace the next nucleotide, but just in the interest of time, I'm going to say that's all I want to trace right now. Take this trace and build in the full backbone. Just going to region it so that you can see what's happening once it starts to do the coordinate calculation. And if I click on build backbone, so it's right below the accept nucleotide button, RCrane going to start calculating out coordinates that match. First it will predict conformers, and then it's going to calculate out coordinates that match the predicted conformers and the predicted sugar pucker, and match the base and phosphate positions that we just built in. Well, this is coming through on the webcast, but you can see basically it looks like a standard Coot minimization. You can see atoms moving around. It's going to try to build one nucleotide at a time closely, and if you have a good enough frame rate, you can see it's going to build a nucleotide and then all of a sudden flash and try to rebuild the same nucleotide. What's happening is it's trying to build in that nucleotide with an anti-sugar, then the same nucleotide with a syn sugar, and then if none of those work, it'll try to put in a high anti configuration, basically just making sure that the minimization doesn't get trapped in a local minima. Minimization is going to take a while since this laptop is rather somewhat slow. Excuse me. The RCrane window will tell your progress. And I should point out that during this minimization, the bases and the phosphates that we just traced in are allowed to move, but only slightly. So you may see things as bases. The base plane may get tweaked a little bit just to help the backbone fit into position, but nothing should go float off, and certainly nothing should get pulled out of density. This should just take another 30 seconds while it does its minimizing. I can on my desktop, which is significantly faster. This whole process is done in maybe 5 to 10 seconds.
One question from the audience here: the minimization you're actually doing here on the screen, is it using the same type of calculation as Coot would use to regularize?
Yeah. So it's the same minimizer that's built into Coot. The idea is basically the same as the regularizement, where I pay attention to density, but very, very slightly. My weight on density is very, very slight. And opposing the torsions from the predicted conformers is the main difference over just sort of doing a standard Coot real refinement. As I'm imposing fairly light torsion constraints, and also impose very, very strict restraints to try to keep the sugars pucker appropriately. So I'm building in either a standard C3' or standard C2' endo sugar pucker, ensure that those don't get flattened or don't get distorted during the minimization. The sugar ring torsions are very, very tightly constrained.
And is it possible to change this weight? Is that something that could be tweaked to the user? Or
not right. Well, you can the source code itself, but right now there's no easy way to tweak those things. I've tried to set parameters that work that are fairly general. The time you can run into problems is if you're trying to build and there's really, really absolutely no density there. Things get pulled a little bit out. But the only reason I was even able to start tracing something there was because I knew exactly what was supposed to be there, because it was back to an experimental map where I had no density published structure, so I knew what was there. For the most part, I've tested a handful of maps, and the seem to work fairly well. If anybody has trouble with them, they're certainly more than welcome to email me and let me know what's happening. I can try to fix it and try to play with weights and see if there's anything I can do to get things working.
Okay. And if this is now partially built, and let's say you want to go and tweak one of the bases, can you easily rerun the refinement in Crane? Or how?
So I can show you later how you'd go about re-rising. Right now, once it's built, there's no way to go back and at least directly change phosphate and base coordinates while it's building. You can go later and fix things. What you can do is tweak the backbone conformers, which is, you can see the window popped up now. We'll let you do that. Now there's no simple way after it's built to go back and tweak those things. And it's some way to go back and tweak a couple things along those lines.
Great. There's also questions from the audience. There was another question, if you could at some point go back and repeat to show people how to place this first phosphate.
Sure, I can do that.
Okay.
I'm just planning on doing that in a minute anyway for something else. So you can see right now, I have the RCrane window. It looks different than it did during the tracing. So right now you can see all suites that got built. So there's 4 suites that we built, 5 nucleotides, but because of that offset thing, we only built 4 suites. You can see conformers that got predicted and built. Suite, so right now we're focused on suite 2. That's what you can see right, that's in the center of the screen. So that got built in as a 1A conformer was the first guess. Before 1A is the helical conformer, so that's usually a pretty safe guess if that's anywhere on your prediction list. If for reason you weren't happy with the way this nucleotide fit into density, you can pick an alternate conformer. And we'll repeat this minimization procedure, not for the entire structure, but just for the affected nucleotides, rebuild them. And so it'll take a minute because this laptop is fairly slow. Since I mentioned before, suites overlap in their central sugar, so if at any point you pick a conformer that requires a sugar pucker change, RCrane wind up rebuilding more than just these 2 nucleotides involved in this suite. It's going to adjust things to try to fix it so that the conformers work, because obviously if one suite ends with a C2' endo sugar pucker, the next suite has to begin with a C2' endo sugar pucker because it's, it's referring to the same sugar. This will just take another 30 seconds while it does the minimization. Yeah, here that's been pulled just a tiny bit, doing the minimization, and there we go. So now we look at the newly calculated suite. So this is now a 1L. That took a while, so I don't really remember what the 1 looked like. Once it has been calculated, you can just jump back and forth between the 2 of them to compare them easily. So it looks fine. I'll stick with that. Notice that in this overview window, I can jump to any suite I want by either using the next suite and previous suite buttons or clicking on it in the overview window. You'll see 5 is highlighted in yellow. So this is using the same stoplight coloring that you get, say, from refinement results in Coot, where if they're in white are sort of standard, those are probably okay. Things that are in yellow, you probably want to at least take a quick look at. I don't have any here, but if I jump back over slides for a second, you can see occasionally you'll get things in orange or red. Again, it means that you probably need to pay more attention than yellow, and red means something probably is going on. You really should take a look at that nucleotide. Two reasons you can get things colored. If I jump over to nucleotide 5, the reasons is if my first and second conformer predictions have a similar score. So these scores, this entire column is going to add up to 1 for every single suite. You can have a 0.25 and a 0.54. So they're not that close, but they are a little close. So that's an indication that maybe you want to take a look at this suite and maybe try some of the alternate conformers. You may get something colored in is if coordinate calculation couldn't find a good set of coordinates that matched both the predicted conformer and the phosphate and base coordinates. So you have singles wrong in the backbone. You may have some bond lengths that are a little bit off. Basically, you should, if the structure looks wrong, you can try an alternate conformer. And very often, it typically means that something went wrong in your tracing. You have either a base or a phosphate that's very far out, but that's flagged for you.
And the score itself, is it calculated based on the geometry or the density fit, or both?
Purely geometry.
Purely geometry.
Yeah. So it's calculated based on a mapping between the pseudotorsions and the backbone conformers, and based on the p-perp distance for how well it matches the sugar pucker.
What's acceptable score?
Typically, the best scoring the best scoring conformer is your best bet to go with. I mean, the suite was flagged because there were things with similar scores. So usually, if something's within, say, a factor of 2, it's at least worth checking it to see if maybe the 1C fits the density better. Density is not great, so it's going to be hard to tell. But your highest ranked conformer is your best bet, regardless of what the score is. These are always scaled so that they always add up to 1. So it's hard to get meaning from the absolute numeric value. It just basically tells you whether or not there was a close second guess, or I think it would jump back over to here. You see that the top score is 1, the second score is 0, so there's really no reason to try any alternate conformers here. Whereas you might want to try things because it's close to an order to a factor of 2 difference. Here again, 1A was the helical conformer, so you might also want to take a look at the 1A conformer here. Just since that's helical, that's sort of a special case. Depending measured, about 70% of your structure should be 1A. So if there's something right in the middle of the helix and everything looks fairly helical, even when 1A is your second or third choice, it's worth at least taking a look and seeing how that fits the data. Okay, so I can just click done. They'll close the RCrane window. And now if I go over to the Coot manager, you see I have RCrane molecule 2. I was tracing something before we started, but you can see that the molecule that we just traced is in Coot's display manager, and it's now a standard Coot molecule. You can do anything you want to it. You can save it, you can delete it, you can change the representation of it, or you copy it into an existing molecule if you already have something traced. You can trace and show how to pick the central phosphate again. So I'm doing that. So to build the first phosphate, the first thing you do is you drag your Coot screen center. So that's this little pink dot. Just drag it somewhere near where you think there's a phosphate. And then go to RCrane menu and select New Trace. You trace in either direction. So this time, before we trace 5' to 3', here I can try this trace 3' to 5'. Pop up. Default is going to highlight its predicted phosphate closest to your screen center. So it's guessing this is what I meant. I put the little pink pointer over here. If that's not right, you can click on the next or previous buttons, and it's going to jump around. So all these green crosses are potential phosphate locations. I can use in previous to move the orange square around. I can also click manually adjust if it didn't find the phosphate you were looking for. And then when I'm done, I just click accept. And now we'll use that phosphate as a starting point for a nucleotide. I shall again. So here I'm going to trace 3' to 5'. So I was actually hoping that RCrane was going to go down, which it didn't. It went up instead. So again, use the phosphate button, trying to build in the correct nucleotide that I was looking for. I jump back here for a second. So here's where RCrane started off with. It's tracing in the wrong direction. You can see this base picking up base pairing with something over here. There's a helix running in this direction. So you can see right now this base is running parallel to the helical axis. That's because RCrane is trying to trace bases in that are standard anti-configuration, and right now we're trying to trace backward and trace the wrong direction. A good way to tell that you're going in the wrong direction is your bases are going to be about 90 degrees off. So if I click the switch button, all of a sudden this base plane looks far more normal. It's picking base pairing with the thing over here that I know it's supposed to be pairing with. Trace the whole helix. See that this being appropriately positioned relative to the rest of the helix. You're tracing, and you need to keep clicking on base, manually adjust, and adjusting by about 90 degrees. It's a good sign tracing in the wrong direction. Just a little, everybody winds up building something. You don't want to wind up tracing the wrong direction. Click delete trace. Thing you can do with RCrane. I'm going to pull up the published structure again. This is a molecule. I didn't build this with RCrane. But if, say, I just had a molecule that I just finished running refinement on, just finished running through mole probity, and there's a section of the molecule that doesn't match the density, or that mole probity has told me that I have sugar pucker errors or steric clashes, I can use the rotomerize existing structure feature. So it's just like real space refinement or regularizement within Coot. Just pick first suite that you want to rotomerize and the second suite that you want to rotomerize. The RCrane window popping up again, and now it's going to rebuild that portion of the structure. So as I said before, if you mole probity told you that something has a wrong sugar pucker, it's actually typically very difficult to try to go in and correct that manually. But here, RCrane should take care of all of those errors and build a proper bone structure for you. This in a minute, but when it finishes, you'll get the exact same review suite window we saw before. It'll tell you what conformers it built in. It'll give you a chance to try alternate conformers, and any suites for you that it had problems building, indicating that either try an alternate conformer in those cases, or maybe there's something wrong with the base or phosphate positioning. Jump PowerPoint slides. I think that's about all I had. Yeah, much the main features of RCrane. I'm still working development of it, so I'd love to hear feedback, either bugs that people are having, or more hopefully suggestions for new features that people would like to see. I have a couple of things planned that I hope will get released soon. And Piotr mentioned there's a PNAS paper that describes the methodology behind this plugin. I'm working on another paper now that will describe more the plugin itself and some of the improvements in the methodology that we made since that original publication. Yeah, but I'll look for questions while this finishes rotomerizing.
So a couple questions from the audience. People were asking about the alternative conformations. If it's something that you could accommodate.
So alternate location indicators.
Right. Yeah.
Right now, Coot can't. RCrane can't. If you rotomerize something with an alternative conformer, alternate conformation in there, RCrane will give you an error message. Build one, you'd probably have to, I guess, trace it twice and then manually tweak things so that one is an alternate conformation for the other. Can work on, but at least right now, unfortunately, no is the answer.
Okay.
Write that down.
And the question was, can you change directions of model building? Is it something that it's you building 3-5 versus 5-3? Can you change the direction as you're building the?
So right now, it only allows you to just do a switch while you're building the first nucleotide. Part of the problem with doing a switch later is it's going to try to reposition all the bases. So there's at least no easy way within RCrane to go back and sort of fix it if it guesses wrong for how to reposition those bases. So right now, if you realize 3 nucleotides in that you are tracing in the wrong direction, you have to go back to nucleotides and then click the switch button and then retrace the other 3.
Okay. And this plot you showed that earlier on, is it something that is available right now within Coot, right?
No, so you can't do this within Coot. You can do this within Amigos 2. So I'm almost positive that's also available through SBGrid, Java application where you can load in an existing crystal structure. I've been told that it is available. You can load in an existing crystal structure and it'll generate a Ramachandran plot for you. You can also use it to search for motifs. So there's certain signatures within the eta theta plot that you can use to find motifs. So there's a couple of features in there. Yes, should be available on SBGrid. So I think if you just type in Amigos 2 from an SBGrid command line, you should get that up and running, and then you can load in crystal structures.
Okay.
And do searches.
Okay. Any other questions from the audience?
I should say rotomerization is finished. Again, really old laptop.
Okay.
But yeah, as you can see, it gives you pretty much the exact same window. Now it's telling you, just rotomerize suite number 368 and 369. Both of these look good, so they're both color 8. And if you wanted to, you could select alternate conformers. You can also, I should point, the original coordinates before rotomerize are now shown in light yellow. The Coot coloring is the new coordinates that are built in. If you want, you can turn off the old coordinates to make the new coordinates a little bit easier to see. You can see it really changed things too much because this section of the structure is pretty good, but had some horrendous star clashes in the backbone, or if you had some sugar pucker errors, they would have been corrected.
If you have any questions, you can use the chat window in the events control panel to type it right now. But if there is no questions, thank you again for coming here today. And if you can put the slide maybe from the presentation. Again, the next seminar, Monday, December 12th at 4 o'clock. Jad from the Lawrence Berkeley Lab will talk about Phoenix. And thank you everybody for participation. Thank you for coming, and have a good weekend. Bye.